Chapter 12, Section 2, Applying Mendel's Principles. Learning objectives. Explain how probability can be used to predict inherited traits. Explain how alleles segregate when more than one gene is involved. Identify Mendel's contributions to our understanding of genetics. Nothing in life is certain. There's a great deal of wisdom in that old saying that genetics is a fine example. If a parent carries two different alleles for a certain gene, we can't be sure which of those alleles will be inherited by any one of the parent's offspring. However, think carefully about the nature of inheritance, and you'll see that even if we can't predict the exact future, we can do something almost as useful. We can figure out the odds. Whenever Mendel performed a cross with pea plants, he carefully categorized and counted the offspring. He had plenty of data to analyze. Upon analyzing his data, Mendel realized that the principles of probability could be used to explain the results of his genetic crosses. Probability is the likelihood that a particular event will occur. As an example, consider an ordinary event such as flipping a coin. There are two possible outcomes of the event. The coin may land on heads or it may land on tails. The chance or probability of either outcome is equal. Therefore, the probability that a single coin flip will land heads up is 1 chance in 2. This amounts to 1/2 or 50%. If you flip a coin 3 times in a row, what is the probability that it will land heads up every time? Each coin flip is an independent event with a probability of 1/2. Therefore, the probability of flipping 3 heads in a row is 1/2 times 1/2 times 1/2 equals 1/8. Remember, each flip is an independent event. The way in which alleles segregate during gamete formation is every bit as random as a coin flip. Therefore, the principles of probability can be used to predict the outcomes of genetic crosses. Like the coin toss, each F2 gamete has a 1 in 2 or 1/2 chance of carrying a small g allele, or a recessive g allele. There are 2 gametes, so the probability of both gametes carrying the small g or recessive g allele is 1/2 times 1/2 equals 1/4. In other words, roughly 1/4 of the F2 offspring should have yellow pods, and the remaining 3/4 should have green pods. This predicted ratio, 3 offspring exhibiting the dominant trait and 1 offspring exhibiting the recessive trait, showed up consistently in Mendel's experiments. For each of his 7 crosses, about 3/4 of the plants showed the trait controlled by the dominant allele, and about 1/4 showed the trait controlled by the recessive allele. Segregation did occur according to Mendel's model. The F2 generation, not all organisms with the same characteristics have the same alleles. Both the big G, or dominant G, and big G, little g, or dominant and recessive G, allele combinations resulted in green pods. Organisms that have 2 identical alleles for a particular gene, like big G, or little g, are said to be homozygous. Organisms that have 2 different alleles for the same gene, for example, big G, are said to be heterozygous. Probabilities predict the average outcomes of a large number of events. One of Mendel's most revolutionary insights followed directly from his observations of the F1 crosses. Every organism has a genetic makeup as well as a set of observable characteristics. All of the pea plants with green pods have the same phenotype, or the physical trait that they looked like. They did not, however, have the same genotype, or genetic trait. Genotypes for us are represented by letters. One of the best ways to predict the outcome of a genetic cross is with a diagram known as a Punnett square. Punnett squares use mathematical probability to help predict the genotype and phenotype combinations in genetic crosses. Constructing a Punnett square is fairly easy. You begin with a square, then following the principle of segregation, all possible combinations of alleles in the gametes produced by one parent are written along the top edge of the square. The other parent's alleles are then segregated along the left edge. Next, every possible genotype is written inside the boxes within the square. After showing the alleles segregate during the formation of gametes, Mendel wondered if the segregation of one pair of alleles affects another pair. For example, does the gene that determined the shape of the seed affect the gene for seed color? To find out, Mendel followed two different genes as they passed from one generation to the next. Because it involved two different genes, Mendel's experiment is known as two-factor or dihybrid crosses. Single gene crosses are known as monohybrid crosses. Next, Mendel crossed the F1 plants to produce F2 offspring. Remember, each F1 plant was formed by the fusion of a gamete carrying the dominant big R, big Y alleles with another gamete carrying the recessive little r, little y alleles. Would the two dominant alleles always stay together, or would they segregate independently so that any combination of alleles was possible? In Mendel's experiment, the F2 plants produced 556 seeds. Mendel noted that 315 of the seeds were round and yellow, while another 32 seeds were wrinkled and green. The two parental phenotypes, however, 209 seeds had combinations of phenotypes and therefore combinations of alleles that were not found in either parent. This clearly meant that the alleles for seed shape segregated independently from those from seed color. Put another way, genes that segregate independently, such as the genes for seed shape and seed color, do not influence each other's inheritance. Mendel's experimental results were very close to a 9:3 to 3:1 ratio. Mendel had discovered the principle of independent assortment. The principle of independent assortment states that genes for different traits can segregate independently during the formation of gametes. Independent assortment helps account for the many genetic variations observed in plants and animals and other organisms, even when they have the same parents. Mendel's principles of segregation and independent assortment can be observed through one- and two-factor crosses. Mendel's principles of heredity, observed through patterns of inheritance, form the basis of modern genetics. These principles of heredity are as follows. The inheritance of biological characteristics are determined by individual units called genes, which are passed from parents to offspring. Where two or more forms of alleles of a gene for a single trait exist, some alleles may be dominant and others may be recessive. In most sexually reproducing organisms, each adult has two copies of each gene, one from each parent. These genes segregate from each other when gametes are formed. Alleles from different genes usually segregate independently from each other. Mendel's principles don't apply only to plants. At the beginning of the 1900s, the American geneticist Thomas Hunt Morgan wanted to use a model organism of another kind to advance the study of genetics. He decided to work with tiny insects that kept showing up, uninvited, in his laboratory. The insect was the common fruit fly, the Drosophila. Drosophila can produce many offspring. A single pair can produce hundreds of young. Before long, Morgan and other biologists had tested all of Mendel's principles and learned that they applied to flies and other organisms as well. In fact, Mendel's basic principles of inheritance can even be used to study the inheritance of human traits and genetic disorders such as cystic fibrosis.